Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Wyatt Technology, I'd like to welcome you to Crystallized Biotherapeutics, optimizing monoclonal antibody crystallization using light scattering. My name is Elizabeth Lamb, and I'll be the host and the moderator for today's event. I'd like to introduce our presenters for today. First is Lynette Schroeder-Tedesco, Group Leader, Crystalomics for Ajinomoto Althea Incorporated. And our second presenter is Jonathan Alvarez, Research Associate 3, also with Ajinomoto Althea. So with that, I will turn the presenter ball over to today's presenters.
Thank you for the introduction. I'm happy to be here today to talk about dynamic light scattering and how we use it to rapidly determine crystallization conditions for biotherapeutics here at Ajinomoto Althea. I'll talk about biotherapeutics and crystallomics and give a brief overview. Then I'll talk about dynamic light scattering and how we use it in protein crystallization. Jonathan Alvarez will talk about the case studies we performed where we looked at Herceptin and Rituxan. And then we'll give a brief conclusion and summary of our talk. After that, we will try to answer your questions, and we'll get to as many as we can before the time is up. Biotherapeutics and crystallomics. So biotherapeutics is the largest growing class of drugs in development. Some estimates put the rate of growth of the number of biotherapies in development at 25% a year. They are used to treat a wide range of conditions, including cancer, leukemia, and autoimmune disorders. Some of the benefits of these therapies is that there is a low incidence of side effects with low toxicity, and they are a highly specific treatment. These therapies do come with some challenges, though. They often require very large dosage volumes, which means IV administration. And I don't think I know anybody who enjoys being at the doctor's office with a needle stuck in their arm, waiting around. As drug formulators have worked towards making higher concentration formulations to address some of these issues, they often come into hurdles and roadblocks with the stability and viscosity. Our crystallomics technology can address some of these issues. So what is crystallomics? It's a portfolio of patents and intellectual property surrounding protein crystallization and formulation that we seek to address these issues. We're able to crystallize proteins, peptides, and antibodies, and then take these crystallization conditions from the very early research stage, nanoliter volumes, where we first discover the conditions, and scale them up to a manufacturing volume greater than 5 liters. We can formulate these crystallized drug substances into drug products, and we have the technology to fill vials or prefilled syringes with the crystalline drug product suspensions following GMP guidelines. One of the challenges I mentioned was viscosity. And to demonstrate how our technology can address the viscosity issue, we crystallized the commercial protein in infliximab and compared the viscosity of the soluble formulation against our crystalline formulation. Now you can see with the blue diamonds that after about 100 mg per ml, the viscosity of the soluble protein increases very rapidly, whereas the viscosity response to concentration of the crystallized protein remains low, and even at 200 mg per ml is under 50 centipoise. With these capability of getting to higher concentrations, we are able to lower the dosage volumes. And 3 commercial proteins that we developed in order to demonstrate our technology were infliximab, rituximab, and trastuzumab. In each case with these antibodies, we were able to lower the dosage volume to under 2.5 mils, which would make these drug products plausible to administer with a subcutaneous injection instead of an IV. With rituximab, we were able to lower the dosage volume from 75 mils down to 2.3 mils. So with our Crystallomics technology, we're able to offer our clients a second-generation drug product, thereby extending their patents. We're also able to achieve stable, high-concentration protein formulations of up to 350 mg per mil, thereby decreasing the dosage volumes while maintaining low viscosity with good injectability and low initial burst. The crystals can also be complexed, creating sustained release formulations. These sustained release formulations could possibly decrease daily injections to weekly or monthly injections with a stable PK profile. Another application of the Crystallomics technology would be for protein purification, and by adding the protein crystallization to the manufacturing process, we're able to eliminate or minimize the need for the protein A purification and end up with a highly pure product. So how do we get these benefits? Well, the very first step is to discover the crystallization conditions. Now, protein crystallization can be really tough. It's usually a very trial and error sort of process where you usually screen your protein against hundreds, if not thousands, of different conditions using different concentrations and pHs, buffers and precipitants, and it's actually really tough. In the past, it's taken us 1 to 1.5 years to develop crystallization conditions for new antibodies, which, if you think about the development timeline for soluble therapeutics, from discovery to the initial animal studies, toxicology and animal studies can be about 9 months. So if it's taking us a year to a year and a half to even get to the point where we could start the formulation development process, we are way behind, and it makes our technology not very competitive. So what can we do to improve our crystallization rate? Well, we've turned to dynamic light scattering with a high throughput characterization of the protein in multiple buffers to dramatically improve our timeline. I'm going to take a step back now and talk about dynamic light scattering. So what does dynamic light scattering do? Well, it measures the Brownian motion of small particles in solution by analyzing the changes of scattered light as it passes through the sample. The fluctuations to the light intensity can be used to calculate the diffusion coefficient and the hydrodynamic radius. So a little more about the mechanics of the DLS. A laser is shot through a polarizer and into the sample. The light is scattered by the particles and is measured on a detector. As the particles move and oscillate, this causes the intensity of the scattered light to fluctuate. At short time delays, the intensity correlation is high because the particles haven't moved much. As the time delay becomes longer, the correlation decays, and due to the destructive and constructive forces of the oscillation of the wavelengths. Smaller particles move faster and have faster decay rates, whereas larger particles move more slowly and the decay rate is longer. After an autocorrelation analysis produces these decay rates, you can calculate the diffusion coefficients, which can then be used with the Stokes-Einstein relationship to calculate the hydrodynamic radius of the molecule. This information can be used to calculate the size information of the particles in the solution. Now here on this graph, they have plotted, or they are showing the correlation of the decay rate of VSA, comparing it to 50 nanometer latex spheres. as well as a combination of BSA and the latex spheres. You can see that the decay functions for each of these are different. You can accurately quantitate the polydispersity based off the width of the average radius up to 30%. On this slide, it demonstrates the utility of the hydrodynamic radius. So when you have a monodispersed particle size, your hydrodynamic radius will have a very narrow width. It'll be a nice tight peak. As you start to get dimers and trimers and more aggregates, you will end up with a more polydispersed sample, and the width of your hydrodynamic radius will start to get much larger. Now, if you have a significantly polydispersed sample, such as multiple proteins or very large aggregates, you may actually get a fully multimodal sample, although not often do you see completely disparate peaks, and I'll talk about that in a second. So here, with the regularization analysis of the size distributions for BSA, the BSA alone is the red peak, and you can see it has a nice narrow peak. The 50 nanometer latex spheres is a slightly wider size distribution, and it's also much bigger. And when you combine the BSA and the latex spheres together, you are able to completely resolve both peaks and see the two peaks. One thing to remember about this, though, is that it is a low resolution method, and you need to have a difference, 3 to 5 times difference in radii in order to resolve these peaks. Most of the time, what we see here is just a broadening of the width of the size distribution. So what can you measure with DLS? You get size information, and you get changes in the average radius. You can get a polydispersity, so the width of the radii and the average radius, and you can resolve populations that differ by a factor of 3 to 5 times. You can also get a percent mass for species up to 200 nanometers. Here at Ajinomoto Althea, we use the DynaPro DLS Plate Reader 2, and we usually use a 384 microwell plate. It's capable of measuring particles that have a size between 0.5 and 1,000 nanometers, with a sensitivity down to 0.1 mg per mil of a 14 kilodalton protein. One feature that we like is the temperature control, because temperature can be an important factor with protein crystallization. So that allows us to look at how the protein responds at different temperatures, as well as allowing us to do the initial melting studies to see how stable the protein is in the different buffers. So how does dynamic light scattering apply to protein crystallization? And this slide really just to impress upon what is protein crystallization used for. Primarily, it's used for structure determination. This is the main way that people determine the structure of the proteins, and this can be used to develop targets or drugs and better understand how the studied protein fits into the bigger picture. It can also be used for drug product formulation and protein purification. So again, back to the question of how do we get proteins to crystallize? It's actually not an easy feat. As I mentioned earlier, it's a largely trial and error process. But some of the things we do know is that we want our protein to be homogeneous. So we want it to be folded consistently. We don't want it to be, some of the proteins this way and some of the proteins another way. We want the proteins to be able to come together and assemble into ordered lattices, and we want the protein to be in the metastable zone, which means not too soluble and not aggregated. We also, for our uses, want the protein to be stable in the chosen conditions, so we don't want the protein to degrade while we're crystallizing it and formulating it. That would be counterproductive. we need our protein to still be active when it is administered and dissolves into the soluble protein. So it is quite a balancing act to get all the conditions just right. The factors influencing crystallization are the protein concentration, the ionic strength of the solution it's in, as well as the salt content. The pH can influence how the protein is folded, the conformation of the protein and the tertiary structure. And in general, as you approach the pI of the protein, you decrease the solubility of the protein, and that is something we do take advantage of and can definitely influence the crystallization. Another factor to consider is the temperature. You don't want your protein to unfold or to aggregate, and so, but some proteins will only crystallize at very specific temperatures. So it's definitely something we look at. Now, this picture down below just gives you an example of some of the phase diagrams that we usually map at the very beginning of our projects. At least if we're lucky, we get to see that crystallization window kind of in the beginning of our batch development process. The red wells indicate aggregate protein. The crystals are that happy window where the proteins will crystallize. And then the clear wells are the conditions where the protein stays soluble and does not crystallize. So what criteria do we look at when we're using the DLS for buffer selection? We look at percent polydispersity, and that tells us whether the protein is homogeneous or whether it's starting to aggregate. We look at the hydrodynamic radius, and that gives us information about how the protein is folded and whether it is starting to open up. We look at the melting temperature to see that the protein is stable, and that gives us an indication later on stability, on potential stability issues. And we also look at the diffusion coefficient, and that tells us whether the protein molecules are attracted and likely to aggregate or whether they are repulsed by each other and will tend to stay in solution. For our very initial screens, we focus on percent polydispersity and diffusion coefficient. Our workflow looks at a little something like this. We usually evaluate 8 buffers, 8 formulation or 8 crystallization buffers at one time at 4 different concentrations. Each protein buffer sample at 4 protein concentrations. The diffusion coefficient is calculated by looking at the diffusion at 4 different protein concentrations. We measure each sample in triplicate, and that leads to 1 full 384 well plate. Now, most of our clients are ready to develop these proteins into therapeutics, so protein quantity is not much of an issue, although we do try to be careful with the proteins. It takes us about 2.5 hours to prep the plates, including the buffer exchange using our robots. And then the way we have our DLS set up, it takes about 6 hours for the DLS plate to be read. And that usually happens overnight. So when we come in the morning, we have all of our data ready to evaluate. Back to percent polydispersity. A polydispersity of less than 15 or 20%, depending upon which source you want to quote, is correlated with a monodispersed sample. A polydispersed sample would be anything definitely over 20%. We usually try to target less than 15% for our conditions, though. And unfortunately, a monodispersed protein is not a guarantee of successful crystallization. However, a polydispersed sample is a fairly good indication that you won't get crystallization in those conditions. So below we have an example of the same protein in 10 millimolar tris at pH 8. It gave us a percent polydispersity of 9.9. The very same antibody in 20 millimolar heaps at pH 7 gave us a percent polydispersity of 21.9. When we looked at the KD, you can see that a positive KD is where the molecules are repulsed from each other. So they don't really want to be close to each other. They aren't attractive. They're unlikely to cause aggregates. And when you're doing soluble formulations, that's a good thing. It's also a good thing in our case. A negative KD indicates that the molecules are attracted to each other and likely to aggregate and form non-ordered aggregates and particles. So again, these are results where we looked at the same antibody. The first sample was in 5 millimolar bistress, pH 6. You can see it has a positive KD, indicating the molecules were not attracted to each other. The same antibody, when placed in a 30 millimolar histidine buffer at pH 6, gave a negative attraction, so those particles would tend to aggregate and come together. Now we usually look at the positive KD, but we want a very slightly positive diffusion coefficient. That way we can control the attraction and the rate at which the molecules come together during crystallization with the addition of other precipitants and by varying the conditions. Jonathan Alvarez will now take over to talk about the case studies of mAb crystallization.
Thank you, Lynette, for the introduction. The first case study that I'd like to speak about is with Herceptin. Herceptin is a commercially available mAb, quite expensive, but we were able to purchase some for the use here at Ajinomoto Althea. So utilizing that pre-crystallization method that Lynette already detailed in the above slides, we were able to analyze a percent polydispersity and KD value of Herceptin in a variety of different buffers, 32 to be exact. So after the analysis using the dynamic software, we were then able to rank these different buffers depending on their percent PD and KD values. First and foremost, we look at the percent PD. Anything above 20% is an automatic no. We cast that out. We don't even consider it because we need a monodispersed protein in whatever buffer that we're looking at. So anything 20% or 19 and below, really that gets a green light, and we're able to move forward with the next parameter. And that next parameter is that KD value. As Lynette pointed out, we are really looking for something that is more slightly positive, so just above that 0 mark. We'll also consider things that are slightly negative, but the reason why we're more interested in the positive KD value is because that's indicative of a repulsive molecule in the buffer system, whereas the negative KD value is indicative of an attractive molecule in that buffer system. So if we can have that positive buffer, we have more control when we get to the crystallization methods. And another thing that I'd like to point out is that the table that we're looking at here, these top 10 buffers, this is the table for T1. And T1 is the results 24 hours after the buffer exchange process. Our ranking system takes into account T1 and T0, T0 being immediately after the buffer exchange. And we're interested in these two time points because we want to see if there's any change in the percent PD or the KD values in that matter of 24 hours. This is important to us because when we're talking about setting up our crystallization experiments, we could buffer exchange one day, set up experiments the next day. We want to make sure that whatever the protein is suspended in, it's going to be stable enough to be able to at least run 24 hours later in our crystallization screens. So those are just some two factors that we consider. So we compared the T0 results to the T1 results, and then make an educated decision based off of those two ranking systems. So here, after comparing the T0 and T1, we were able to go ahead and move forward with the #1 ranked buffer, 30 millimolar Bistris, pH 6.5. So here we're looking at comparing the results of our crystallization screens of the proteins in two different buffers. The first buffer is the formulation buffer that the drug product came in. The second buffer is that 30 millimolar Bistris, pH 6.5, that we selected after putting the protein through our pre-crystallization methodology. The protein crystallization screens were all the same for both of these different buffer types. They were incubated at 23 degrees, both for 6 weeks after that buffer exchange process for the Bistris. So we can compare the crystallization hits, and that's what we're really interested in. Do we see any significant changes in the number of crystal hits before or after our high throughput protein buffer exchange process? And as you can see here in our formulation graph, the formulation buffer graph, there were 27 crystal hits found in those crystallization screens. And we can come then over to the right graph where we're looking at the protein that was suspended in the 30 millimolar Bistris, pH 6.5, and we have an increase to 48 different crystal hits in that new buffer system. So close to 50% increase in the number of crystal hits found utilizing the pre-crystallization methodology. This is significant for us, especially when we're talking about scaling up or selecting crystal hits in specific crystallization conditions that will help us in the long run, especially developing our formulation buffers and that crystallization suspension that is ready to be a drug product. So here are some crystal hit examples of what they look like. You can see that some of these, there's only 2 here out of those 48. But it's just something that we want to show. You know, everybody likes to look at crystals. Pretty pictures are, again, also awesome. So here we have needle-like crystals that we were able to produce using that 30 millimolar Bistris, pH 6.5 buffer system for Herceptin. And this is by no means anything ready for our batch development, nor is it ready for a structural determination project. A crystal hit is exactly that. It's just a hit. It's something that you can optimize, something that you can work towards to produce that perfect crystal for your structure determination, or for, in our case, that crystal that we need in order to move forward with our batch development. So just, again, pretty crystals. I like to look at them. I'd like to share that with you. So that led us to the actual formulation development of Herceptin. And so as you can see here, we're comparing now the commercial drug product versus our formulated crystal suspension. And as you can see, there are some pretty significant changes, differences between these two. The drug product concentration that's commercially available is 21 mg per mil with a dosage volume of 13 milliliters, while our formulated crystal suspension is 350 milligrams per milliliter at a volume of 0.8 milliliters. So definitely significant differences that we can see here, something that we're very proud of, just, you know, something that our technology can really help in terms of patient comfort, in terms of, you know, even the time and effort a patient has to spend in a clinic. So something that we've been able to improve with our crystallization suspensions. The second case study that I'd like to speak about is with Rituxan. So we put Rituxan through the exact same methodology that we did with Herceptin, the differences being in the buffers that we selected for the process. Each protein is going to have the buffers tuned specifically towards that protein PI. So the buffers are not always going to be the same. However, the process is going to be the same. So again, looking at T0, so the results and the data immediately after the buffer exchange, as well as 24 hours later at T1, we're able to rank and choose our buffer depending on those mean percent polydispersity and KD results. You're looking at the ranked buffers from T1. Again, I want to point out, we use both T0 results data as well as T1 results and data in order to determine the most suitable protein buffer. In the last case with Herceptin, we were able to go ahead and select the number 1 ranked buffer system. In this case, however, with Rituxan, the number 7 ranked buffer system was chosen, 10 millimolar histidine, pH 6.5. Again, that goes back to any changes that were found between T0 and T1. So we take the whole thing into consideration when making our choice to move forward. So 10 millimolar histidine, pH 6.5 was slightly positive in both T0 and T1. It had great percent polydispersity, so we decided to move forward with that. So here are the results after the 6 weeks incubation time. These results are comparing the Rituxan suspended in the formulation buffer that's commercially available and the Rituxan suspended in the 10 millimolar histidine, pH 6.5 that we chose based on this pre-crystallization method. As you can see, there were 66 different hits, crystal hits, when the protein was in the formulation buffer. And then you can compare that to close to 200 crystal hits when the protein was suspended in our 10 millimolar histidine, pH 6.5. I calculated it out, it's close to 200% increase in the number of hits found using our methodology. So just astronomical difference, really gives us a large amount of crystal hits that we can choose, different conditions that we can choose when we're moving forward in our batch development. Here are some examples of some crystal hits found from the Rituxan experiments. Again, these are nothing that's ready to go and just throw in a formulation right away. We would use these hits and optimize them so that we can get the crystals that we desire when it is that we're wanting to move forward as our batch scale increases. Lastly, so comparing the commercial drug product versus our formulated crystal suspension, again, we see a dramatic increase in the protein concentration from the commercial drug product of 10 milligrams per milliliter all the way up to 320 milligrams per milliliter. And then that decrease of 75 milliliter dose volume down to 2.3 milliliter dose volume. So significant changes that are happening with the crystallization formulation suspensions, changes that we're really proud of. So to sum up, the few things that we'd like to point out, again, just reiterating the difficulty of actually crystallizing mAbs, especially free mAbs. All the mAbs that we work on here in Althea have been free mAbs, so nothing bound to any antigen. So very difficult process, and yet we've been able to do it successfully utilizing our DLS. Just to kind of point out where we were before utilizing the DLS, we were working on some projects that it would take a year and a half to just get one single crystal hit. And so now, after implementing the DLS in our methodology, we're seeing dozens, if not hundreds, of crystal hits in a matter of 6 weeks or less. And we've already proven that with over 7 different protein molecules. This is saving us time, this is saving us effort, and lots of headaches. Our walls are definitely doing much better now without any head banging going on. So we're really pleased with our specific instrument, the DynaPro Plate Reader 2 that we purchased from Wyatt. It's definitely been that strong man in the lab in our recent projects. Here's a slide of our references. If you are interested, we can send them off to Wyatt and you can contact them that way, or you can just pause the webinar right now and take a closer look. We'd like to acknowledge our Crystalomics team here at Ajinomoto Althea. From our lab techs to our scientists to our managers, everyone has just been awesome and provides great contribution to all of our efforts. And it's so great to work with each and every one of them. very happy to just be able to come into work every day, and that's because we have such a great team. And we'd also like to thank you, the listener. We appreciate you paying attention through this whole webinar. And lastly, we'd like to thank Wyatt for not only their instruments, but also for giving us this platform to speak today. Without them, we'd still be waiting on a year and a half before we get any crystallization hits. So thank you, Wyatt. And at this time, we'd like to take any of the questions that you may have. So Lynette and I are here to answer any of those. Thank you again.
Thank you very much, Jonathan and Lynette. Yes, we do have some questions, so let's get started with them. Our first question is from Allison. For crystallized drug product formulations, do you see an increase in soluble and insoluble aggregates upon dissolution of the suspension?
So that would definitely be a concern, I think, with any drug product formulation. It can vary from product to product. In general, though, we optimize away from seeing any additional aggregate. And in some cases, we actually see better aggregate profiles after the crystallization than we do with an equivalent soluble formulation.
All right. Another question. This one is from Michelle. You mentioned monoclonal antibodies as traditionally difficult proteins to crystallize. Typical antibodies are IgG structures with only 2% carbohydrate. What about more heavily glycosylated proteins? Are these more difficult to crystallize? And can you provide an example of one that was crystallized?
Yeah, so just to mention that here in the lab, we haven't worked with anything really heavily glycosylated, so I can't give, personal example that we've worked on. But literature does show that it can also be a little difficult to crystallize these heavily glycosylated proteins, especially when we're looking at crystal structures, because you really need to make sure that you're looking at homogeneous protein samples. So the glycosylation can cause some issues with that. A quick literature search did show some examples of some crystallized glycosylated proteins, and I can share that. Ramaswamy et al. In the IUCRJ journal crystallized a glycosylated protein from Diploratera punctata, and that was just in 2016. So a quick literature search will give some more examples of that. Unfortunately, just here in the lab, we haven't worked on any of those heavily glycosylated proteins.
Oh, sorry. I was just going to add in that according to the literature, the approach for crystallization would be very similar for the glycosylated proteins as it is for the antibodies. So in general, you look for conditions giving you monodispersed, homogeneous proteins, folding and structure, and then you do a screen with a diverse set of conditions looking to see what sort of crystals, if any, you can generate.
All right, our next questions are from Eric. After the buffer is chosen, what are the differences in conditions that produced crystallization hits versus misses?
Yeah, so that actually, they're going to differ a lot. It really depends on the protein, because you never know exactly which crystallization conditions are going to produce crystals. So when you ask how different they are, they can vary in terms of the precipitant. The different molecular wet pegs can be different. The precipitant can be a salt instead of a peg. And then the buffers themselves that you're looking at in crystallization conditions can vary as well from protein to protein. So just because we've used the DLS to identify which buffer is most suitable to suspend the protein in, that doesn't zone in, or that doesn't hone in onto a particular crystallization condition.
All right, the follow-up question from Eric. How important is the crystal morphology in the ranking of conditions that produced hit?
I would say, well, the morphology, especially when we're moving forward in our batch systems, is important. We normally, because we're going to be utilizing some type of syringe system for the most part, something that has a lot of needles or clusters and, that kind of morphology is going to be more difficult to push through a syringe as opposed to something more, square or cubical or even round like a rice or football kind of shaped crystal. So morphology is important, and we try and do our best to optimize the morphology when moving forward. With crystals, I mean, sometimes you just got to go with the hand that you're dealt. We do have, for example, our HT crystal formulation is a more needle-like crystal, so we can get it to work. It's just one of those things that we consider. If we can optimize it early on, then we try and do that.
All right, our next question is from Hang Jun. What about the PK profile? Did you get a longer half-life?
So that does depend a little bit on the drug product. For the PK profile, we have had studies done with HGH, and I believe those results were published a while back. The studies were done a few years ago. And what we do see is we actually get a slightly better PK profile, and that for uncomplexed formulations, it takes a little bit longer for it to enter the blood serum because it takes a few minutes for the crystal to dissolve, typically 10 to 15 minutes. Now, other drug products that we've had, like the HGH, were complex, and so those would dissolve and enter the bloodstream over the course of a week. So it does depend on the product, but typically we would expect uncomplexed crystals to dissolve within about 15 minutes.
All right, our next question is from RK. What solution conditions actually promote crystallization of proteins other than the pH, salt, and protein concentrations?
So when we're looking for our protein buffer suspension, or our protein suspension in that buffer, we're not looking at any other type of parameters besides pH and buffer concentration. However, when we get into the actual crystallization methods, the temperature is going to be an issue. You already mentioned your pH concentration, but then your precipitant concentration and type are also going to influence your crystallization as well. Yeah. So for example, our pegs, we use a lot of different pegs. Peg smears, even what we're utilizing in the lab. So a lot of these different things influence the crystallization of the protein.
Our next question is from Lee. How do you actually confirm it's a, quote, crystal hit, close quote, other than from simple visual inspection? Many times you get crystals from small molecules or particles that look like crystals, but are actually pegs.
It's a great question. It's definitely always something that we think about. Definitely we have, our imaging system has UV imaging capability. So the first thing that we do is test with that. If we do have some doubts from our UV imaging, then we'll also use is-it dye, so we'll dye the crystals. And then we'll even use poke test, so we'll manually poke the crystals and determine that way as well. And the last thing that we do, especially when we have more sample to work with or more crystal suspension to work with, is that we send them off for X-ray diffraction. So we look for a preliminary X-ray diffraction result, and that really gets rid of any doubt or suspicion that we might have whether something is crystalline or protein, or salt or protein.
Excellent. Our next question. After ranking the formulations by high throughput dynamic light scattering, you select only one to go forward into crystallization trials. Are you missing a potentially much better formulation by picking only one, or do you expect fairly similar performance from the top-ranked formulations according to DLS?
Yeah, so what we didn't mention was that we actually test two different protein buffers after our ranking system. And again, that's because we want to make sure that we're not missing any, or we're not missing, another buffer that can possibly give us better data. And so we put it through what we call a concentration optimization test after this initial ranking system. And this concentration optimization test, we use that to help us determine which of the two best buffers is more suitable, as well as which concentration of the protein is more suitable for our crystallization screening process. So again, just to sum, from these ranking results using the DLS, we select two different buffers that we then put through that concentration optimization test, where we then, after that, select our one final protein buffer suspension and use that for our crystal screens.
You mentioned that the DLS measurements of a 96-well plate takes 6.5 hours, which comes out to about 4 minutes per well. Pretty long for DLS. Is that because you're measuring at multiple temperatures, or because you scan the entire plate multiple times at a fixed temperature? And what's the rationale behind this?
We actually use a 384 well plate for this process. So that would, bring it down to about a minute per well. We can definitely optimize a little bit more, I think, in terms of how many images or how many measurements we take, and then how long each measurement is. So it really depends too on your protein, and the sample that you're looking at. So right now with the 384 well plate, so I think we do have room for some optimization to decrease that from 6.5 hours. But I think it's still pretty quick, being able to test all those different samples in that amount of time.
Would it be feasible to run the DLS screen with a regular cuvette-based DLS instrument?
So I'll answer that one. And we could get the same measurement with a cuvette, but we wouldn't have the throughput capability. So we wouldn't be able to collect as much data or as quickly determine the conditions, which in practice would probably limit the number of different buffers and conditions we look at. Usually to get the diffusion coefficient, you want to look at multiple concentrations of your sample or of the protein. And so there's a minimum of probably 4 or 5 samples right there with the different protein concentrations. Even at a couple minutes each, when you have to prepare your sample and then wash the cuvette after the reading, that would end up being a lot of time. So in addition to saving the amount of protein that you're using, the 384 well plate is the way to go for us.
Our next question. It sounds like you have a lot of data to analyze after an overnight DLS run. How much of that is automated, and how much net time do you spend processing and analyzing the data? Do you use any tools outside of the software that comes with the DynaPro plate reader?
So the analysis portion actually does take a few hours. So when we first got this up and running, I was looking at around even close to maybe 8 hours of analysis and really digging through the data. But now that we've done it for so long, it's become a lot more proficient. So we're looking at, just a few hours of data analysis, but then that also comes with being able to put that data into a format that we can visualize. So all in all, we're looking something at right now close to about 5 or 6 hours of analysis and creating graphs and things where we can actually make some decisions based off of the data. However, what we're implementing now, actually fairly recently, we're utilizing the statistical analysis program called JUMP, as well as MATLAB. So utilizing both of these softwares, we're able to create or write code to be able to pull out and mine the data for us, and then so MATLAB will pull out, mine the data, and get it all organized for us, and then JUMP we can utilize to create the graphs and to be able to visualize or put it into the visual format so we can make our decisions that way. So our computer guy here on our team basically tells me that what would take 5-6 hours can now take a matter of seconds. So it's going to be really awesome when we finally validate that and move forward with it.
Thank you. We have just a few questions left. Are there any downsides to crystallized high-concentration biologics, whether physiological concerns or product efficacy, shelf life, or manufacturability? Is there any concern that subcutaneous injection of crystals causes cellular damage or rupture?
So we've had our HGH product successfully complete phase 2 clinical trials that was in development. Altus was the original company that promoted this crystallization technology, and they were bringing the HGH product through clinical trials. When they closed their business and were sold to Althea, they stopped the trials and didn't start phase 3, even though they were approved to. But from those studies and from other studies, we don't see any additional tissue damage from the crystalline suspensions. It is equivalent to any of the soluble formulations. So I think safety-wise, the crystals don't pose a problem. There are the additional challenges of developing the batch crystallization conditions as well as developing the formulations, I think is mostly, I believe, because fewer people are doing it, is a little more tricky and novel than doing the soluble formulations. There's fewer analytical tools and fewer publications and journals. There's just less of a community doing it to draw upon. But really, I think the concerns are minimal if you're interested in using a crystalline formulation.
Next, we have a comment and a question. Clearly, DLS gives you a major advantage over the antibodies formulation buffer. Would you recommend the HT-DLS screen for general protein crystallization research?
Yeah, I believe that utilizing this technique would help any crystallographer. It doesn't necessarily have to be an antibody either. It can be any type of protein. I think if the crystallographer is able to find what makes the protein happiest in its formulation suspension or buffer suspension, then it'll just mean, I think, quicker success when they go through crystallization screens.
All right, and we're down to our final question. You mentioned that hydrodynamic radius and melting temperature are also of interest in picking the best formulations to go forward. Have you taken these into account in the case studies? And do you measure TM with the DLS plate reader or another method?
Initially, we did not use the melting temperature or hydrodynamic radius in our buffer selections. We're working on developing those, so they were not presented here in the case studies, but I think they will be of a very big interest. We are starting to use those measurements when we're developing. So after the initial screen, as we have fewer candidates that we're looking at and we're developing our batch crystallization conditions, we're finding them very useful. For the most part, we find the DLS to be one of the best tools to do that in the high throughput methodology. We do have a DSC that we can use, but we use that mostly for our crystalline slurries. The sample size is a bit higher, and you need higher protein concentration than you do with the DLS. So we find the DLS to be pretty useful for a lot of different things.
All right, thank you very much, and perfect timing as well. I'd like to thank our presenters today, Jonathan Alvarez and Lynette Schroeder-Tedesco. I'd like to thank Wyatt Technology for sponsoring today. Finally, I'd like to thank those of you who came and spent this time with us. We know you have many things to do and many places to be, so we're glad that you chose to be with us for a while today. So thank you again so much. On behalf of Cambridge Health Tech Institute's Global Web Symposia, have a great day.